Okay, great. All right. So I'm going to talk about cancer, cancer genomes, and treating cancer genomes. So we'll start by I'll give you a little introduction to cancer and what its causes are. I'll talk about cancer in the immune system, how we read the cancer genome, how we can rewrite the cancer genome, and I'll give you some success stories and caveats about this technology. So, cancer is a very, very old disease. It's been with us since the beginning of civilization. There is this great book, actually written by an Indian physician in New York at Memorial Sloan Kettering, called The Emperor of All Maladies, which is a great guide to the history of cancer and its treatment. So the earliest recorded report of cancer is from 2600 before the Common Era in an Egyptian physician called Imhotep, who described what appears to be a breast cancer in a female patient. Another important event was Hippocrates, who in 400 BCE, who named cancer karkinos, or crab, or claws in Greek. And then in 200 AD, the famous European physician Galen described cancer and named it onkos, for swelling. And that gave rise to the term oncology, which is the study of cancer. So cancer is ancient and it's broad. It affects all of us. In Canada, cancer is now the #1 cause of death, before heart disease, diabetes, and all the other diseases. 2 in 5 Canadians will develop cancer in their lifetime. Pretty much everybody will know somebody with cancer. 1 in 4 Canadians will die from cancer, and when you add up all the number of years lost, it's 1.3 million years of lives that are lost every year. Because of the aging population, we're expecting to see a roughly 40% increase in cancer cases over the next 2 decades. So cancer risk increase. The main reason for this increase of cancer is because cancer risk increases with age. This is a chart on the right which shows the rate of cancer in people of different age ranges, and you see it's very low for children, and it peaks at around 67 years of age, and then it slowly declines. If you look at cumulative cases of cancer, though, there's people who develop cancer in their 60s. There are nearly 3,200 cases per 100,000 of people of these ages living in Canada. So cancer is is a tricky disease. It's very hard to treat. The reason for this is it's very complex. It's not a single disease. There are more than 200 different types of cancer. And cancer acts in many ways like a living organism. It evolves. It becomes resistant to treatment. You treat it, it seems to respond, the cancer goes away. And then a few years later, it comes back. And that's because the treatment didn't kill all the cancer cells, and then they came back stronger. It can lie dormant in the body for many, many years. It's developed ways of evading the immune system so that your white blood cells do not recognize the cancer and clear it. It's caused by multiple environmental and genetic causes that are hard to control. We'll talk a little bit about that. And every person's cancer is unique and requires individual diagnosis and treatment. There's no one-size-fits-all treatment for cancer. And finally, cancer, because it strikes so commonly and unexpectedly, is really causes a lot of fear and concern among people. The good news is that fewer Canadians are dying from cancer, even as the rate is increasing. So at this point, 64% of Canadians who have been diagnosed with cancer are expected to survive for 5 years or more after their diagnosis, and many survive and die till ripe old age and die of other causes. And you can see this really dramatically, and it's because of advances in cancer detection and therapy that between 1988 and 2021, cancer death rates have decreased by quite a lot. It's gone down by 37% among men and 22% among women. We're making progress. We wish it could be faster, but we've made immense progress in the last 50 years. So I'm going to now step back and talk about what cancer is, what causes it, how it develops. So, we're going to look first at cancer at the cellular level, and then we'll look at the genome and individual genes. So cancer fundamentally is an uncontrolled growth of cells. Ordinarily, every cell in your body has a program which tells it to divide and multiply a certain number of times to change into its final form, skin cell, an eye cell, a brain cell, and then stop dividing. That's it, they're done. In cancer, there's something goes wrong with the regulation of cell growth, such that instead of stopping on schedule, the cells continue to divide in an uncontrolled way, and they form eventually a tumor. And every single type of cell in your body has a different cancer type that it develops into. You can have cancer of the mouth, of the GI system, of the lymph nodes, of the lungs, of the stomach, and we can go on and on. And each one of those diseases shares some commonalities, some similarities with other cancers, but is unique in its own way. And in fact, each individual's cancer is different from every other cancer that's ever been seen. So what are the causes of cancer? Well, cancers are caused by an accumulation of changes in the genome that result from DNA damage. So we take a normal cell from in someone's body, and over time, it's exposed to radiation, just for example, sunlight, carcinogens, chemicals in the foods that we eat or drink, and there's a large component just of random chance. So any of these things can take normal DNA and damage it, break it into pieces. Now, the cell doesn't like that, and it has a very good system for repairing damaged DNA. But it's not foolproof. And sometimes when the body repairs one of these breaks, it introduces an error. It's not, the DNA is not the same as it should be. It has what's called a mutation, a variation, a change in the composition of the DNA. And when that hits a particular gene that controls growth, you can turn a normal cell into a cancer cell. So we're going to look at this in a little bit more detail. We'll drill down into this a little bit. Here's the structure of DNA. It's a double helix. You've seen this on everything from coffee mugs to T-shirts to cosmetic products. There's a code embedded in the DNA as a series of chemicals named GCTNA, and these together encode all the proteins that make up your body. And everybody has a slightly different DNA code because nobody is exactly alike. So if we turn this on its side and we look at it the way as someone reading the genome would look at it, you can see a series of these chemicals, G, A, T, and C. This would be a normal sequence. And that is recognized by the cell and used to synthesize a normal protein, which is a folded, very small structure that looks something like this. When DNA damage and repair occurs, there can be a change in the DNA. And here we see a T that's been repaired incorrectly, and it's become a C. And this could be from UV radiation or from a carcinogen in the diet, or it could just be something that happened randomly. And as a result of changing this little change, it has changed the normal protein into an abnormal protein. This is subtle, but you can see a slight, these are not identical structures. You can see it's sort of opened, the protein is opened up here a little bit. The blue area is further to the edge. And what this has done in this particular gene, this particular protein, is it's taken a growth factor, something that tells the cell to divide, which is usually regulated so that it's turned off when it's time for the cell not to divide. It's opened up this this area and turned off the part, and turned the protein on and made it so that it can't be regulated anymore. And now this protein is telling the cell, grow, 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 divide, divide, divide. And that has caused the cell to go from a normal cell to becoming a cancer cell. Because the protein is abnormal and because the code to make the protein has changed due to an error in the DNA. So we do cancer genome sequencing in order to get to the root cause of the cancer in each patient, to understand how that will affect how the cancer will behave, and to come up with treatments for that particular cancer. And so the way we do this is we take a piece of tumor and we go through a process in which we're able to read the base pairs out, read out the chemical code, and then we can line up normal and tumor DNA, and we can see that in the tumor DNA there's been a change here. Here's an area that's been changed, and here's another area very close by that's also been changed. And then from that DNA sequence, doctors can determine the type of the cancer, what organ it's from. Sometimes you don't actually know. You have a patient who has multiple tumors scattered around their body and you don't know where it started. And without knowing where it started, you don't know how to treat it. You can tell what exposures might have caused the cancer. Is it asbestos? Is it cigarette smoking? Is it just age? Did the patient have a genetic syndrome that increases their risk for having cancer? They can tell how rapidly the cancer will grow and propose treatments that the tumor is most likely to respond to. And this is something called precision oncology, where by understanding the cause of the cancer and its particular DNA change, you can find drugs that are most likely to be effective for that patient. And in practice, this is how it works. A patient who needs to have their tumor sequenced will come to the hospital. They'll consent to having their DNA analyzed. They'll then go to the operating room to have their tumor taken out or biopsied. Then a piece of the tumor is taken, or the tumor is taken to the pathology department in the hospital, where a technician cuts the specimen up, photographs it, and prepares it for microscopic examination and for DNA analysis. And so typically what happens is that most of the tumor will go to a pathologist who will look at it in the microscope, and they will attempt to determine whether all the tumor was taken out or whether there's still some tumor at the edges that was not taken out. That's very, very important. And then whatever tumor is left over will get frozen and ultimately sent for DNA sequencing. And so how does the sequencing work? Well, you actually have to sequence two things. You have to sequence the tumor's DNA, but you also have to sequence the patient's normal DNA in order to understand what changes occurred, because everybody's DNA is slightly different. So you take the chunk of tumor tissue that I showed you before, and you take a blood sample from the patient, and assuming that the patient does not have leukemia, which is a blood cancer, their blood will be their normal DNA that they were born with. You then chop up and extract DNA from the cells, giving you a tube with a little bit of DNA here. It's actually white when it's purified. And then that goes, there's several steps here that I'm going to skip over, and it goes into a sequencing machine, which is able to read the base pairs one after another, read the code one after another. And so here is a little teeny tiny segment of the patient's normal blood DNA showing a T here, and then it's changed in the tumor to a C. And then taking all the change, there are usually multiple changes in the cancer, and then taking that sequence, with the changes marked in it, this will then go to a compute system, which will compare those changes to all previously known changes in the cancer and interpret them, creating a clinical genetic report. And there are multiple parts of this, but the most important part is that these reports can often identify known changes which will make a patient more sensitive, make the patient's tumor more sensitive to a particular treatment. So, for example, this patient had a change in a gene called EGFR. We won't go into exactly what that is, but that's known to make the patient sensitive to this list of chemotherapy drugs here. Okay. It also tells the doctor that this is it was a lung cancer. Okay. Sometimes it will work the other way. The patient has a mutation in KRAS, and that tells them that they're not likely to benefit from this list of drugs. So it both tells you what drugs you can give, and it tells you what drugs you shouldn't give, because those will cause side effects without benefiting the patient. Okay. Another really exciting technology that's coming along is something called the liquid biopsy. And in the liquid biopsy, you don't even have to take the tumor out or take a biopsy from it in order to learn something about it. The reason for this is that cancer cells frequently invade the blood stream and shed cells into the blood, which break down over time into DNA. And so you have some circulating DNA in the blood. It's called circulating tumor DNA or ctDNA. And if you take a blood sample and you sequence it very, very, very deeply, you can start to see these pieces of tumor DNA. And now you can do exactly what you did before. You can predict what drugs the patient will respond to. You can say something about what caused the cancer and where it's from and what type of cancer it is. But now, instead of having to go to the operating room, patient just goes to a local clinical laboratory and has their blood drawn. And the very exciting thing about this technology is that you can monitor the patient's therapy to see how well it's working just by taking blood, but just by following the blood. So, for example, one thing that ctDNA is now used for is that if a patient is going to go for surgery to have their tumor removed, you really want to understand whether the surgery was successful, whether all the tumor was removed or whether some cells were left over, because those cells will regrow. So what we do is we take a blood sample from the patient before they go to the surgery, and we measure how much tumor DNA is in there. And then the patient goes for the surgery. We wait a couple of weeks for things to settle down, and we take another blood sample and sequence that. If the surgery was successful, we expect the patient's tumor DNA to go down to 0. It will become undetectable. If the you could now watch the patient and not give them further treatment for a while at least. If, however, there's still DNA, tumor DNA in the blood after the surgery, then that patient's surgery was incomplete, and either they need a subsequent surgery, or they need to be treated more aggressively with chemotherapy and radiation therapy. So this allows us to know how the patient is going to do and whether the patient needs additional therapy. You can also continue to take blood tests while the patient every few months and measure if you can see if the tumor DNA is coming back. And if it starts to come back, you know the patient is recurring, and you could start treatment for a potential recurrence or metastasis. And this is less expensive and much more convenient than having a CT or a whole body MRI scan. Okay, so those are the things that we can do with reading the genome. Now let's talk about writing or rewriting the genome. So until recently, the genome has been read-only. We can look at at the changes in it, but we can't really change. We can't conveniently or easily change the DNA, change the DNA itself. But there was a revolution over the last decade called CRISPR, which lets us rewrite the genome very accurately. So CRISPR, it stands for clustered regularly interspaced short palindromic repeats. I don't really want to have to explain all that, but it's a system that was evolved by bacteria as a defense against bacterial viruses. These are things called bacteriophage. So you probably may not have known this, but bacteria, we get viruses, bacteria get viruses too. And the way CRISPR works is when a virus infects a bacterium, it injects a little bit of viral DNA into the bacterium. And the viral DNA, its mission is to take over the bacterium and make it produce more viruses. But in bacteria that have the CRISPR system, they have a combination of an enzyme. This is a protein that's able to digest DNA, and something called CRISPR DNA, which is a little piece of bacterial DNA which recognizes viral DNA. And when the CRISPR DNA recognizes the viral DNA, it activates this enzyme, and this enzyme chops up the viral DNA into lots of little pieces. Chop, chop, chop. And the virus is dead. And now, using molecular biology, we can take that CRISPR system that was invented by bacteria and repurpose it to our own uses. And we can apply it to other types of DNA, including human DNA. We can take any piece of DNA, and by recognizing its sequence, we can chop it up, or we can delete a piece, or we can in fact replace a piece of DNA. So how can that be used to treat cancer? It is very early days. It just started to experiment with this, but it looks very promising, and that's what I'll tell you about. So there are a lot of challenges adapting this bacterial system to working in the human body. First of all, the tumor, it's a big solid mass. And how do you get the CRISPR DNA and its enzyme into the tumor efficiently? And more important, how do you ensure that the CRISPR is going to target the tumor cells and not do any damage to normal cells? And then the further challenge is that every cell in the tumor is actually slightly different. And it may not have the DNA change that the CRISPR system is targeted against. And unless you kill all the cells, some of them will grow back and reform a tumor. So right now, most CRISPR is not directed directly at tumors, but it's directed at enhancing the patient's immune system. So I'm going to tell you some stories about that. So first of all, this may surprise you, the human immune system is actually very good at detecting and deleting early cancers. In fact, the vast majority of potential cancers we never see. They never become a problem because the immune system recognizes them and kills them before the tumor gets large enough for it to cause any symptoms or other problems. And it's only a very small number of cancers that evade this immune mechanism and are able to grow. Here's a picture of here's a series of pictures of a white blood cell, which is blue at the bottom, attacking a cancer cell. It's green at the top. This is using a technique called electron microscopy. And the cell, the white blood cell, recognizes the cancer. It sends out these extensions, which are filled with chemicals that are actually kill cells, and the cancer cell explodes. The way that white blood cells recognize cancer cells is that these cells have a receptor. It's a sensor which is able to recognize abnormal proteins that you were not born with. Cancer cells typically express abnormal proteins, and sometimes they're on the cell surface because of the changes, the mutations in their genome. Once the white blood cell recognizes this abnormal protein, it goes into action. It sends out those extensions, and it kills the cell. So in a patient who has cancer, by definition, that cancer has already evaded the immune system in some way, and the immune system needs some help. And one of the ways that the research community has figured out to augment, to strengthen the patient's immune system is with a system called CAR T cells. In the CAR T cell system, so T cells are a type of white blood cell. They're the main ones that recognize cancer. You take the patient, you collect their blood, you harvest out this particular type of T cell, and then you genetically reprogram it by giving it a new receptor, a new sensor that can recognize the particular type of abnormal protein or proteins that are expressed on that cancer patient's tumor cells. So you've created a supercharged, program-targeted white blood cell. And now in the laboratory, in Petri dishes, you expand these cells, you tell them to divide, so now that you have a whole bunch of them, and you infuse them back into the patient, and the CAR T cells attack the cancer cells and kill them. It's not 100% effective, but it works quite well and can give patients many years of life. There are problems with CAR T cell therapy. One is that the CAR T cells need to be created from the patient's own white blood cells. You can't use someone else's immune cells. You can't go to the blood bank, because those cells will, when they're infused into a different person than the donor, will recognize that the patient's normal cells as being foreign and attack them. So it has to be their own cells. And this is time consuming. It's very expensive to do this for each patient. It costs about $500,000 in Canada for a CAR T cell, for one course of CAR T cell therapy. These patients with cancer are often quite ill, and you may not be able to get many white blood cells from them because they're ill. And the province will only pay for these under very strict qualification guidelines. You basically have to have failed all other therapies. before you're eligible for CAR T-cell therapy. So wouldn't it be great if we could make these CAR T-cells in bulk using blood banked cells from unrelated donors, and have them basically in the refrigerator where it could be used for the next patient who comes in with cancer without having to make them each time from that individual's blood. So that's the first thing that CRISPR therapy has been used for. In 2022, there was this 13-year-old teenage girl, Alyssa Tapley, who had intractable leukemia. She'd gone through all the courses of treatment, and her leukemia had come back. She was too sick to give blood for her own CAR T-cell therapy, so the researchers used CAR T-cells from other donors. And what they did is they took these T-cells and they used CRISPR to knock out 3 of the genes which are involved in recognizing healthy normal cells. So these cells would no longer be able to see Alyssa's own healthy cells as being foreign. They won't attack them. And then they edited a 4th gene to make these white blood cells recognize Alyssa's cancer. They infused it they infused these activated T cells, these CRISPR modified T cells into Alyssa. Within 28 days, she was in remission. The cancer completely disappeared. And in this year, 2.5 years later, she's still free of leukemia. She seems to have been cured, although that in cancer, that's never a guarantee. So this is very promising therapy. It's being done on an experimental basis in clinical trials in the UK, not currently available in Canada. But if it continues to be continues to work in more individuals, someday it will be available here. Another approach that people have taken, a very similar one, is to take the patient's white blood cells and make them more aggressive. So in addition to the foreign protein receptor, white blood cells also have a receptor, a sensor, for a signal that's on the surface of many normal cells, which is a "don't kill me" signal. It tells the white blood cell, look, this is a normal, healthy cell. Don't kill me. Leave me alone. Go somewhere else. Now, cancer cells, of course, have evolved in many cases to also express this signal. So that's one of the reasons that the patient's immune system, without modification, is unable to recognize and kill the cancer cell. One approach to overcoming this type of resistance is something called a checkpoint inhibitor. This is a drug that you take orally or by injection, which partially inactivates the "don't kill me" receptor and allows this white blood cell to activate and kill the cancer cell. Another approach to this, however, is to combine CAR T-cell therapy with checkpoint inhibitor therapy. So what you do is you take the patient's blood, as we did before. You add this recognition sensor for the cancer, but you also do one more thing. You take the CRISPR system and you knock out the white blood cells "don't kill me" receptor. So now it's a supercharged white blood cell that will go after the cancer and ignore the fact that the cancer is sending the signal. Yeah, so yeah, here we've knocked it out, and now we have a hyperactive white blood cell. And so this technique is showing a lot of promise and is being used in multiple clinical trials in Europe, in the States, and in some and I believe there are some trials in BC for advanced colorectal cancer, leukemia, kidney cancer, and breast cancer. These are just in the very early phases where they're testing safety. If it works, it will be another way of applying CRISPR to cancer care. And then ultimately, at some point in the future, it would be, we would like to be able to apply gene editing directly to the cancer cell in order to kill it, either by chopping up its genome or by correcting the errors in the genome and returning the cell to a normal state so it stops dividing. This has been done in cancer cells growing in petri dishes, and where the cells are easily accessible and you can get the CRISPR into them easily. It's a challenge to get working in living models, and people are trying it with mice only at this point. It's not nearly at the stage where you could try it on human beings. And some early successes have been using the same technology that was used in the COVID vaccine by putting the reagents inside these tiny little oil droplets that are able to penetrate into cells and release their release their contents. So stay tuned to that. This may someday become a real therapy. So that's pretty much all I wanted to say here. The take-home message is that cancer genome sequencing, in which we read the cancer genome, it's a powerful technique for precision oncology. It's being used right now to improve cancer care. Writing the cancer genome with gene editing tools holds promise, but right now the near-term promise is to improve cancer immunotherapy by modifying the patient's immune cells or modifying unrelated donors' immune cells so that they can attack cancers. And many challenges still at this time need to overcome in order to apply this technology directly to cancer cells. Thank you, and I am happy to take any questions.